Good evening. I'm Professor Jo Robinson, Head of the School of English Literature, Language and Linguistics. I'm delighted to welcome you all to the 2021 Fickling Lecture on Developments in Children's Literature, which is presented in association with 7 Stories and the Children's Literature Unit in the School of English Literature, Language and Linguistics at Newcastle University. Tonight's lecture is entitled What is Life? And is presented by Sir Paul Nurse. This evening's prestigious lecture is an annual event funded by David Fickling Books, one of the UK's few independent publishing houses. David Fickling is one of the most adventurous and successful British publishers for children. He's responsible, for instance, for Philip Pullman's His Dark Materials trilogy and the prequel, The Book of Dust. As well as enjoying the books, many of us will have watched the first 2 seasons of the BBC adaptations. The final season was commissioned in December, so we can hope to see that next year, COVID allowing. David Fickling has also published Demon Voices, a book of Pullman's essays, including one he gave to launch the Fickling Lecture series. Other David Fickling book successes are Mark Haddon's The Curious Incident of the Dog in the Nighttime, and The Phoenix. This wonderful weekly story comic has helped a great many children get through the endless days of lockdown. One of his most recent innovations are books explaining some of the large areas of science and discovery. Paul Nurse's What is Life has been described by Bill Bryson as a nearly perfect guide to the wonder and complexity of existence. I'll be back after the lecture for a live Q&A with the speaker. So if you'd like to submit a question, please do so via the YouTube live chat box on your screen or via Twitter at InsightsNCL. If you're tweeting about the event and you want to share your thoughts, please use the hashtag InsightsNCL. And now, here to introduce tonight's speaker is Professor Chris Day, Vice-Chancellor and President of Newcastle University. Thank you.
Good evening, everyone, and a very warm welcome to the 2021 Insights Fickling Lecture. This evening's speaker really is a man who needs no introduction. Knighted by the Queen in 1999 for his contributions to cell biology and cancer research, a Nobel Prize-winning geneticist and Director and Chief Executive of the Francis Crick Institute, a former Head of Cancer Research UK, and President of the Royal Society from 2010 to 2015, Paul Nurse is rightly lauded as one of the leading scientists of our time. His discoveries have helped to explain how a cell controls its cycle of growth and division. The broader significance of these findings lies in the knowledge that errors in cell growth and division may lead to cancer and other serious diseases. Throughout his remarkable career, Paul has been the recipient of more prestigious awards and honours than it is possible to recount here. But notable among them, along with the Nobel Prize, he shares with Leyland Hartwell and Tim Hunt for their discoveries of protein molecules that control the division of cells in the cell cycle. He was awarded the French Légion d'honneur in 2002, the Royal Society Copley Medal in 2005, and the Japanese Order of the Rising Sun in 2018. I've known Paul for many years as a medical scientist and head of our Faculty of Medical Sciences, and now as Vice-Chancellor. He has been a source of great personal inspiration for me, and we are privileged to call him a loyal friend and supporter of Newcastle University. He is a member of our distinguished family of honorary graduates, and in 2011 he performed the opening of our Badley Clark building, the home of our Centre for Bacterial Cell Biology. So it is my great pleasure to welcome Sir Paul Nurse to deliver the Fickling Lecture on developments in children's literature. Paul's lecture this evening is entitled, What is Life? In his talk, he will be discussing 5 great ideas of biology, from the cell to the logic of life. The lecture takes its title from Paul's book, published last September, in which he takes up the challenge of defining life in a way that every reader can understand. Brian Cox has described it as a beautifully written exploration of perhaps the most important question in science. Praise indeed, and I can think of no better note on which to invite Sir Paul Nurse to take the stage. Thank you, Paul, for presenting this evening's talk, and thanks to you, our audience, for joining us. I hope you enjoy the lecture.
When David Fickling asked me to give the Fickling lecture on developments in children's literature, I was rather taken by surprise. It is true that my home in Oxford is full of children's literature, but that is because my wife, Anne, is passionate about children's books. We have more than 10,000 of them. But David explained he wanted me to speak about a book I had written called What is Life, because he knew I had written it with an interested 13 to 14-year-old teenager in mind, and so he viewed it as non-fiction children's literature. So I accepted his proposal, and here I am. But to give the lecture a clear children's literature flavour, I thought I would start with some relevant quotations from one of the world's greatest writers for young adults, Philip Pullman. My book, What is Life, tries to answer the question of what links together all the extraordinary diversity of life on our planet, as well as with possible life elsewhere in the universe. Now, Philip Pullman extends this even further when he describes imaginary life forms in his novel, The Amber Spyglass, the third part of his Dark Materials trilogy. And I'm going to start the lecture by reading to you a section from The Amber Spyglass about these imaginary life forms. This begins with the scientist Mary stepping through into another world, where she has discovered extraordinary animals and plants like ours, but which are also very different. These include wheeled animals, or apparently wheeled animals, and gigantic trees with huge seed pods. I pick up the story as Mary spots a herd of strange grazing creatures. She moved on and found herself getting closer to a herd of those grazing creatures she'd seen the previous evening and whose movement had puzzled her without her knowing why. They were about the size of a deer or antelopes and similarly coloured, but what made her stop still and rub her eyes was the arrangement of their legs. They grew in a diamond formation, 2 in the centre, 1 at the front, 1 under the tail, so that the animals moved with a curious rocking motion. Mary longed to examine a skeleton and see how the structure worked. For their part, the grazing creatures regarded her with mild, curious eyes, showing no alarm. She would have loved to go closer and take time to look at them, but it was getting hot, and the shade of the great trees looked inviting. The closer she got, the more astounded she was by the enormous size of the trunks, as wide, she estimated, as the house she lived in, and as tall as she couldn't even make a guess. Feeling too hot to move any further, she lay down to rest between the roots of one of the giant trees, with her head on her rucksack, and fell into a doze. Her eyes were closed for 20 minutes or so, and she was not quite asleep when suddenly, from very close by, there came a resounding crash that shook the ground. Then came another. Alarmed, Mary sat up, gathered her wits, and saw a movement which resolved itself into a round object about a yard across, rolling along the ground, coming to a halt and falling on its side. And then another fell further off. She saw the massive thing descend and watched it crash into the buttress-like root of the nearest trunk and then roll away. What were they? Seed pods? Then she heard a low thunder-like rumble, which was hard to locate until she saw a cloud of dust moving along one of the roads, towards the stand of trees and towards her. It was about a mile away, but it wasn't moving slowly. What she saw made her head spin. At first, it looked like a motorcycle gang. Then she thought it was a herd of wheeled animals, But that was impossible. No animal could have wheels. She wasn't seeing it, but she was. There were a dozen or so. They were roughly the same size as the grazing creatures, but leaner and grey-coloured, with horned heads and short trunks, like elephants. They had the same diamond-shaped structure as the grazers, but somehow they had evolved on their fore and rear single legs a wheel. But wheels do not exist in nature, her mind insisted. They couldn't. You needed an axle with a bearing that was completely separate from the rotating part. It couldn't happen. It was impossible. Then, as they came to a halt about 50 yards away and the dust settled, she suddenly made the connection, and she couldn't help laughing out loud with a little cough of delight. The wheels were seed pods, perfectly round, immensely hard and light. They couldn't have been designed better. The creatures hooked a claw through the centre of the pods with their front and rear legs, and used their two lateral legs to push against the ground and move along. While she marvelled at this, she was also a little anxious, for their horns looked formidably sharp, and even at this distance she could see intelligence and curiosity in their gaze. The nearest creature touched its own breast with its trunk and spoke. Was it 3 syllables or 2? The creature spoke again, and this time Mary tried hard to make the same sounds. Mulefa, she said tentatively. Others repeated Mulefa in her voice, laughing, and even seemed to be teasing the creature who had spoken. Mulefa, they said again, as if it was a fine joke. Thank you very much, Philip, for that. These Mulefas are life forms, albeit imagined ones. And in this lecture, I'm going to explain what is a life form. It's one of the fundamental questions in biology, an easy question to ask, but not so easy to answer. There is an extraordinary diversity of life on our planet. All around us are bacteria, fungi, plants and animals, including, of course, ourselves, human beings. The living organisms found on Earth are physical entities that maintain themselves, grow, self-organize and reproduce. So what links all these different life forms together? I think it was a butterfly, shown here, that first started me thinking seriously about questions like this. It was early spring. I was perhaps 12 or 13 years old and sitting in the garden when a quivering yellow butterfly flew over the fence. It turned, hovered, and briefly settled. Then a shadow disturbed it and it took flight again, disappearing over the opposite fence. That intricate, perfectly formed, brimstone yellow butterfly made me think. It was both utterly different to me and yet somehow familiar too. Like me, it was so obviously alive. It could move, it could sense, it could respond. It seemed so full of purpose. I found myself wondering, what does it really mean to be alive? In short, what is life? This question has been asked by others for years. For example, this is the title page of a book also called What is Life? It was written in 1944 by the famous physicist Erwin Schrödinger. His book was influential, including for me when I first read it, now nearly 50 years ago. My approach to that question which Schrödinger asked was a little different from him. It's to consider 5 important ideas of biology, describing where they came from, explaining what they are, then pulling them together to develop principles fundamental to life. The first of these ideas is the concept of the cell. The cell is the basic structural and functional unit of all life on our planet. It is life's atom. Cells were discovered by Robert Hooke in 1665. He used a razor to cut slivers of cork and examined them under an early microscope. He saw neat arrays of boxes he called cells after the Latin word cella for small cubicle, shown here in his drawing published in a book called Biography. A few years later, the Dutch draper Antony van Leeuwenhoek, working in Delft in the Netherlands, scraped between his teeth and discovered single-celled life, bacteria. He sent this drawing, shown here, to the Royal Society in London, and it has a charming image of a bacterium doing a loop-the-loop under the microscope, shown here in Fig C to D. He was rather disturbed by this discovery of microorganisms because he was very proud of his dental hygiene. He was a close neighbour in Delft to the painter Vermeer, who painted two pictures of a scientist. Here is one of them. I like to think this is Leeuwenhoek, but have to admit to you there is no good evidence for this speculation. Although when Vermeer died, Leeuwenhoek was the executor of his will. Over the next 2 centuries, it became clear that all living organisms consist either of a single cell or of a collection of cells. This conclusion was summarized by the German zoologist Theodor Schwamm, who in 1839 wrote, "All organisms are composed of essentially like parts, namely of cells." As well as cells being the basic structural unit of life, they are also the basic functional unit of life. This was expressed by another German, the pathologist Rudolf Virchow, who in 1858 wrote that the cell is a vital unit bearing the complete characteristics of life. He also said, "All cells come from cells, omnius cellula acellular." This means that cells arise through the division of pre-existing cells. So cells are interesting. But if I have failed to convince you of that, let me show you this picture of a sperm or many sperm and an egg. And remind all of you that you were all once a single cell, just like this fertilized egg. Cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. This means understanding cells is key to understanding life, so they will figure prominently in what I have to say. A cell is a bounded physical entity, separated from its environment, surrounded by a semi-permeable lipid membrane, which separates them from their surroundings, but allows communication with those surroundings. This separation allows order to be built up inside cells at the expense of increasing disorder outside cells. In this way, life does not contravene the second law of thermodynamics, which states that the universe as a whole moves towards disorder. This is often noted by physicists thinking about life, like Schrödinger, as seen in this quote from his book. He was astonished that life could escape the decay into atomic chaos, especially from one generation to the next in living organisms. And it led him to propose that life had a code script. These quotes shown here introduce the second idea, based particularly on code script, that I want to share with you. This is the idea of the gene as the basis of heredity, which had its origins with the work of the Augustinian monk Gregor Mendel, abbot of Brunnow Monastery. He tackled the problem of inheritance by carrying out crosses of pea plants which had different characteristics, and analysing the outcomes in thousands of the progeny formed in these crosses. He did this work and published it in 1865. I found this picture of his garden in 1981, when I first visited his monastery in the middle of the Cold War. Mendel crossed peas of varied characteristics, including plants of different heights, different flower colours, and different seed shapes, as shown here. He famously observed simple ratios in these characteristics in the progeny produced by these crosses, ratios like 3:1. This eventually led to Mendelism, the idea that heredity is based on indivisible particles or factors inherited through the germ cells, the pollen and ovules in plants, sperm and eggs in animals. These factors were found to be linked to the chromosomes that were discovered a little later to be separating in dividing cells, as shown here in this late 19th century drawing of onion root tip cells. These Mendelian hereditary factors are what we now call genes. In the 1940s, Oswald Avery in Rockefeller University, New York, of which I was once president, showed genes were made of deoxyribonucleic acid, or DNA. The structure of DNA as a double helix was established about 10 years later in London by Rosalind Franklin, Ryan Gosling and Maurice Wilkins, and in Cambridge by Francis Crick and Jim Watson. Crick and Watson famously argued that the double helix structure of DNA could explain heredity through the linear sequence of bases making up DNA and the pairing between complementing bases, adenine pairing with thymine and cytosine with guanine. This is all shown on this picture here. This double helix is like a twisted ladder. The rungs of the ladder are made up of links between those complementing bases. When these links are split, two templates are formed which are used to make two new identical helices as shown on this slide. This explains how the DNA is copied during cell reproduction, accounting for heredity. It also explains Schrödinger's code script and the permanence of the genes between generations, because DNA acts as a code script, which is precisely copied every time a cell divides. The sequence of bases encoding the genetic information is kept stable in the middle of the DNA polymer double helix. Crick went on to propose what is called the central dogma, explaining how DNA encodes the structure of another cellular component, the proteins. The central dogma describes how the sequence of bases making up a gene is copied from the DNA to ribonucleic acid, or RNA. This RNA acts as a messenger between the DNA located in the nucleus, where the chromosomes are, and the cytoplasm, where the proteins are made. The sequences of the messenger RNA determines the structure of proteins, and it was shown there is one gene for each protein. These discoveries were made in the second half of the 20th century, and they laid the foundations for molecular biology. They are critical for understanding life. Central to them is the genetic code. Heredity is written in this 4-letter code made up of the 4 bases, adenine, thymine, cytosine, and guanine, with the information stored in a linear form. This way of storing information is the same as sentences in a book, the words in this talk, or bytes in a computer. It means that the linear digital storage of information was invented by life over 3 billion years before the computer age. The nature and function of the gene introduces the next two ideas I want to talk about: life as chemistry and life as information. First, life as chemistry. The functioning of living organisms is based on chemical activity and physical forces. The origins of these ideas can be traced back to French chemists. After initial work by Lavoisier, the father of chemistry, it was Louis Pasteur who carried out experiments on sugar beet fermentation in the north of France, which began the concept that life should be viewed as chemistry. Pasteur was analyzing the fermentation of sugar beet, which was used to make alcohol, and he was doing it at the request of industrialists near where he lived, because the fermentations sometimes failed and made acid instead. He demonstrated successful production of alcohol required the activity of a single-celled living organism, yeast. He proposed that the yeast cell produced substances that made alcohol from the sugar derived from the sugar beet. And if yeast was not there, but bacteria instead, then the fermentation made acid instead of alcohol. Then Pasteur brilliantly concluded, more generally, that fermentation was a physiological act yielding products for the cell. Chemical reactions are an expression of the life of a cell, is what he said. In other words, life is chemistry. But it is a very elaborate and special chemistry based on carbon polymers. We have already met the carbon polymers that make up the nucleic acids, DNA and RNA. And now we need to consider the carbon polymers that make up proteins. The substances from yeast that Pasteur postulated made the alcohol are proteins that act as enzymes which catalyze chemical reactions. Proteins are polymers of amino acids based mainly on carbon and nitrogen. The carbon atoms form both the backbone to the polymer and also the links to the side chains of the amino acids. The structure of such a polymer is shown here, or at least part of such a polymer. The different amino acid side chains are chemically different. Some are positively charged, some negatively charged, some repel water, some are big, some are small. Examples are shown here of the 20 different amino acids that make up the proteins, with these different coloured side chains. The result of this is a wide variety of chemistry that can produce elaborate structures which carry out many and varied chemical reactions. This contrasts with DNA, which is stable and is not chemically diverse. In fact, it's chemically rather dull. The chains of amino acids making up protein polymers fold up in many ways to make complex molecular structures. I show here a protein change and a three-dimensional molecular structure derived from that one-dimensional chain. These varied structures, combined with the varied chemistries of the 20 different amino acids, accounts for the wide diversity of chemical reactions that enzymes can carry out. Enzymes and other proteins act as chemical machines. These machines produce the huge range of chemicals required for living organisms to function. They make new molecules, they break molecules down, they recycle them, and they generate large molecular assemblies which synthesize polymers of nucleic acid, protein, lipid, and carbohydrate. These chemical machines can also, some of them at least, do physical work. They make motors, which carry cargoes around the cell on protein-based trackways. They act in hybrid ways, combining chemistry and physics, capturing energy from sunlight or breaking down sugars to produce energy-rich chemicals that drive the activities carried out in cells. All this activity means that there are thousands of chemical reactions being carried out simultaneously and close to each other in living cells. A small sample of these are shown as dots on this map of the central metabolism of cell, part of the chemistry of life. Because these reactions require different chemical conditions, they have to be separated from each other. This is achieved by compartmentation, brought about by the surfaces and folds of the enzymes, by complexes of several enzymes acting together, and by membrane-bounded organelles visible in the cell here, and schematized in this diagram shown here. This compartmentation consists of many different chemical microenvironments, which allow the hugely elaborate chemistry of life to take place. For this to work, all this chemistry must work together as a whole. It has to be organized together, which requires communication between these different chemical microenvironments and compartments. Communication is the transfer of information. And that leads to the next idea, life as information. Life is a complex system, as was first argued by the philosopher Immanuel Kant. The operations of the complex systems of life are based on this management of information. I will give two examples to illustrate this. For the first example, I return again to the structure of DNA. Here is that structure, the double helix replicating, that we've seen before. This structure of DNA is iconic. It is beautiful. But its real beauty lies in the fact that it only makes sense when it is recognized that it is a digital information storage device, one written in that linear script of 4 letters we have already discussed. The linear polymer of DNA is ideal for storing information, with the encoding letter bases turned inside the helix, protected from chemical change. Even better, this information can be turned into chemical action that can do work through the varied chemical activities of the proteins. For me, this is a wonderful characteristic of life, all built on carbon-based polymers, the ability to encode information in one-dimensional linear structures and to carry out chemical and physical work in 3-dimensional structures. The second example of information in life I want to tell you about is regulation. That is, making the complex systems in living organisms act as a whole, with purpose. A familiar example of regulation is found in man-made machines. This example is the governor, developed by James Watt to regulate steam engines. As the engine goes faster, the balls shown here swing out and shut off the steam. And when the engine goes slower, they swing on in and switch on steam. This leads to homeostasis, maintaining a constant speed of the steam engine. The governor is an example of what we call a negative feedback loop, shown here in the context of a metabolic pathway in a cell. The metabolic pathway goes from A to B to C. As C builds up, it inhibits A to B, shutting off the synthesis of C, leading to homeostasis in the level of C. Negative feedback in cells was demonstrated by the French geneticists François Jacob and Jacques Monod, studying gene regulation of sugar metabolism in bacteria. Other control modules lead to positive feedback. In this case, as C builds up, it accelerates A to B and makes more C. This module acts as an irreversible switch. Once on, it stays on. Many such feedbacks and control modules operate in cells, maintaining homeostasis, as we've just seen, generating switches, timers, toggles, and oscillators, translating chemistry into informational modules. These can be linked together to generate even more elaborate routines of informational management and processing. A metaphor for all of this is an electronic circuit of linked modules. The management of life's chemistry by information allows the behavior of the complex system to act as a whole with overall purpose, such as reproducing a cell, making a butterfly, or maintaining our cells. For centuries, the generation of purposeful behaviors was thought to require a designer, a divine creator. It took evolution by natural selection to change this, and this is my final idea. This is a beautiful idea. It is mainly due to Charles Darwin. It has two parts. The first is that life evolves over time, and the second is that a major mechanism for evolution is natural selection. That life evolves was widely discussed in the century before Charles Darwin. For example, by Lamarck and Charles' own grandfather, Erasmus Darwin. Erasmus Darwin, shown here together with Charles, was a colourful character, a scientist, doctor, and poet. In fact, most of his research was written in poems. He was a Republican, a proponent of women's education, and a great conversationalist, despite a terrible stammer. He also argued for the evolution of life, suggesting simple, formless mollusks could evolve into complex animals. The evolution of life implies that there is a tree of life, that living organisms are related. If life only arose and survived once, as seems likely given the conserved chemistry and coding found in all life, then everything on Earth, all life on Earth, is related to each other. This connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859, where he illustrated a tree of life. Charles Darwin gathered evidence in favour of life evolving through natural selection during his trip round the world in Her Majesty's ship, the Beagle. Natural selection is a consequence of populations of species exhibiting variations caused by inheritable genetic changes. These can be caused by mistakes when the DNA is copied, or if DNA is damaged by external causes such as radiation. If these variants influence characteristics that make those organisms more successful, better adapted, and therefore reproducing more, then that particular genetic variation will spread through the population. Over time, such genetic variations will accumulate and lead to changes in the population, such that the individuals of that new population will no longer be able to breed with the original population. This eventually leads to a new species. It is the origin of species, a species which is better adapted and is more successful in survival and reproduction. Darwin came to this view in part by studies of finches on the Galapagos Islands. Some finches had big beaks that could break nuts. Others had thin beaks that could more easily catch insects under the bark of trees. They became adapted to their lifestyle and environment by being specialized better for what they had to do in order to succeed. Just like different pliers shown on the right here are specialized to do different jobs. But the difference between beaks and pliers is that pliers are intelligently designed by human beings for different specialized purposes, whilst the finch's beaks achieved specialized purposes by natural selection. without a divine designer or a creator. Now, for such natural selection to take place, three things are needed. The first is reproduction, the second is a hereditary system, and the third is that that hereditary system needs to exhibit variability upon which selection can work. Let me show you how all this can work based on the ideas of the cell and the gene. Here we have a cell undergoing reproduction. I want you to imagine that it is a single-celled life form and that it has a brown coat, the color of which is determined by a gene in its chromosomes. Imagine when the cell divides and copies its DNA, it makes a mistake. So the cell coat gene is altered and the cell now makes a red coat. Also imagine that red coat cells survive better, perhaps because they do not taste so good to a predator. As a consequence, the red cells replace the brown cells in the population, and evolution by natural selection has taken place. This is a profoundly important idea in biology, so much so that the 20th century geneticist Hermann Müller used it to define what life is. He defined life as living things have properties which allow them to undergo natural selection to evolve. Now, in a couple of minutes, I'm going to consider this further. But before doing that, I want first to say goodbye to Darwin as an old man with the final monumental sentences from his Origin of Species. He ends The Origin of Species with this sentence: "Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." Note how he was relating his work to the great physicist Isaac Newton. He was arguing that biology can have fundamental laws just like physics. Now, before I move to defining life, or trying to define life, I first need to consider the difficult category of intermediate life forms, the viruses. This will help clarify how we should think more about the nature of life. Viruses, such as the one shown here, have a genome based on DNA or RNA. They undergo evolution by natural selection, and thus they pass Müller's test. But they can only reproduce when they are inside the cells of other host living organisms. They do so by hijacking the cell's molecular machinery to copy the virus's genome and to make more viruses. This means that a virus is completely dependent upon another living entity. You could also argue that a virus is alive when it is in a cell of another living entity and is a lifeless chemical when it's outside such cells. So the question is, are viruses truly alive? In thinking about this, it is important to remember that other life forms are also, to a greater or lesser extent, dependent on other living beings. The many parasites that live on or inside the cells or bodies of animals, plants or fungi, are clear examples of this, although the extent of their dependencies are less than it is for a virus. There are certain amino acids that humans cannot make, which we obtain from other living organisms. Even free-living microbes are dependent upon molecules made by other living organisms, albeit simple molecules like glucose and ammonia. Plants are rather more independent, using the energy of the sun to make biomolecules from simple chemicals, including carbon polymers derived from atmospheric carbon dioxide. But even plants rely on bacteria found in their roots that capture the nitrogen from the atmosphere. Perhaps the most independent organisms are the cyanobacteria, single-cell microbes which capture both carbon and nitrogen from the atmosphere, and then energy also from the sun. Therefore, there is a graded spectrum of living organisms, from the viruses through to plants, with a wide range in between. These different life forms have varied dependencies on other life forms. In the case of the virus, that dependency is very strong, whilst in other living organisms, the dependency is weak. But I argue that these different life forms are all alive because they all evolve by natural selection. But it also means that life on Earth is fundamentally connected, both through being related as a result of evolution and also through these deep relationships and interdependencies. I began by saying that by considering some of the important ideas of biology, as I have done, we could establish principles to understand what life is. Let me now summarize where I think we have got to, though I have to warn you that it does not lead to a neat dictionary-like definition of life. We start with living things are independent, bounded physical entities, separated from their environment, in communication with their environment, allowing them to generate order and organization. The basic unit of life on Earth is the cell, and multicellular organisms are aggregates of cells. The core principle of defining life, I believe, is the one developed by Muller. Life forms are entities that can undergo evolution by natural selection. By doing so, they acquire purpose. They are entities that build, maintain, and reproduce themselves. Living things can evolve because they have the following attributes. They reproduce, they have a hereditary system, and that hereditary system exhibits variability. Life forms are chemical, physical, and informational machines. They construct their own metabolism, which is used to build and maintain themselves, to grow and to reproduce. And finally, life certainly on Earth is based on carbon polymer chemistry. These polymers produce nucleic acid-based linear information storage devices and protein-based chemical and physical machines. Integration of all these functions requires the management of information. Inputs are gathered from within and outside the cell. They are processed, stored, and used to instruct cells and organisms to maintain themselves, to grow, and to reproduce. Living things on Earth are related by common descent and are closely interconnected and dependent upon other living things.
Hello? Hello. Thank you, Sir Paul, for that fascinating talk, taking us from the beat of a butterfly's wings to the fundamental questions of life. Sadly, we're all in our separate rooms tonight rather than together here in Newcastle, so we can't all applaud together, but I'm sure that everybody at home is joining me in thanking you for that wonderful talk and also for being here live with us for the Q&A. So our audience has been sending in some questions during the lecture, so I'm going to be asking some of those over the next 15 minutes or so. And I'm going to begin with a question from Mark, who asks, you've already achieved so much in your career. What would you say was your most important discovery?
Well, I have to say, having 2 children, to be perfectly honest, but perhaps that's not what we were really the question was about. I think I worked on the what controls cells dividing from 1 into 2, and that's fundamental to the growth of all living things, including ourselves. And I think probably the discovery that excited me most was working out and identifying the components that controlled that in a very humble little yeast, and then showing that it was exactly the same control from yeast through to humans, and had remained unchanged for 1,500 million years. So much so that I could take the gene from humans, put it into yeast, and it could completely substitute for that gene in yeast. And that just blew my mind and still blows my mind away. So I think that's probably what I rate the most.
Probably blown everybody's minds that's just been listening to you now. So thank you for that. And then a question from Kate as to, is there anybody in your career who you've admired or who has served as an inspiration or a role model to you?
Yes, that's a good question. I mentioned Sidney Brenner in my book, who was a great wit. He died a year or 2 ago and was one of the great biologists of the 20th century. And in addition to that, was very funny. And it really helps to be very intelligent and very funny at the same time. But I also want to mention 2 people who very early in my life, in my 20s, were a great help to me. They were my supervisors in Edinburgh and in Bern, Switzerland. Murdoch Mitchison, who was the son of Naomi Mitchison, the novelist actually, and Urs Leopold. And what I wanted to say about both of those was their generosity. When I was in their labs, I must have published nearly 20 papers, and neither of them would put names on my papers because they said they didn't contribute enough. Huge generosity to a young colleague to help them on their way. So I would like to acknowledge them as well.
Okay, fantastic role models. I've got a question, if I may, and then there's another question about the book. So when David Fickling asked you to write the book, and it's absolutely beautiful for those of you who haven't seen it yet, it's gorgeous, and I really recommend that you get hold of it. When he asked you to do that, what were your challenges in terms of taking these huge ideas and making them accessible, as I think you've done brilliantly?
Well, I think the first thing was, what daunted me was writing all those sentences, each of which required a verb, actually. So that was a bit of a challenge. I don't write too badly, but I have to go over it and over it and over it again. I was hugely helped by my editor, Ben Martinoga, who I also acknowledged, who, if you like, turned me more into a human being, if I can put it that way, and managed to emphasize the sort of more human contacts, and that greatly helped me. So explaining things wasn't so difficult, making it accessible a little bit more difficult, and making scientists look as if they were human was probably the most difficult bit of all.
Okay, so I've got a follow-up question to that from David, and having heard that answer, we may know the other one. So David says, I'm a big fan of the book, and do you have any plans for another one?
Well, I'm not sure. It took me quite a lot of effort to write this one, and I think it is okay. I'm waiting to see on the jury's out. I must admit, I now know it's published in 22 different countries and 19 different languages. So it will have a sort of worldwide sort of circulation. If it works and people like it, then maybe I'll look at another book. I'm not quite sure what it might be. Maybe What is Science, for example, could have a go at that. But let's see if this one is really let's see if it works, and if people like it, I'll think about it.
Okay. I think it's worked. So a question from Sarah as to what you most enjoyed about writing What is Life for a young audience in particular.
Well, I definitely had a young person in mind, not just a young person, because I've written something mainly not for scientists at all. It's for young people who have got an interest in anything really, but an interest in the world, including science, but also for adults who would just like to be introduced to what biologists think life is about. And what I enjoyed I did enjoy weaving in stories from my own research career, because I find that people who aren't scientists just think that we boffins sit down and, there's a great idea every other week and we devise experiments and prove this and that and so on. And it's just all baloney. It's nothing like that at all. It's more like wandering around on a moor in the fog, in a bog, and not knowing where you're going. And then just occasionally the sun lifts the cloud and you suddenly see where you are for a short period of time. You get somewhere and then down it comes again. And I wanted to convey that sort of randomness, that serendipity, and also the joy of it too, but combined with just the hard graft. I can't tell you how many hours you fail to do anything. It's just an incredible amount. To try and convey all of that is what I sort of enjoy, because I sometimes find these sorts of popular books just elevate the scientist onto a pedestal as if they just do everything brilliantly. And I know, because I have been successful, it isn't anything like that.
Okay, great. So that good sense of what it actually is to be a scientist and that experience. So a question from Andrew now, which I'm sure we all really want to know the answer to actually. Was it a surprise when you were awarded the Nobel Prize? Does that come as a bolt from the blue?
It didn't entirely come as a bolt from the blue. I wasn't that old, at least for a biology PhD, a biology researcher. I was 50 years of age, so over 20 years ago. And the reason it wasn't a complete surprise is there's sort of precursor prizes, ones that lead up to it. And I'd won several of those. So I did know I was likely to be on a shortlist. When it happened, I could just say a little story there. I knew what day it was announced because some journalist had phoned me up and said, "Do you think you'll win the Nobel Prize tomorrow?" And I cursed him, of course, because, you don't want to be reminded of this. But I was... The next day, I got no phone call. I went out to an architect's office, actually, because I was helping with getting a museum built to recognize Grigor Mendel, the great founder of genetics, who worked in Brno, now in the Czech Republic. So I was sitting in the architect's office because they were designing it. And I got this phone call, came into the office, and they said, "Could you turn on your mobile phone?" Because I was, over 50, so my mobile phone is constantly off. And when they did that, there was this recorded message with somebody with a hot potato in their mouth. And what I thought, he was Swedish, and what I thought was I was being asked by some other journalist, because I was cursing them at the time, to comment on who had won it this year. So I re-recorded, I listened to it again, and then gradually it dawned on me, they were telling me it was me who had won it. So then I said something really stupid. I went back into the room, and weirdly, Jim Watson was there of Watson and Crick, because he was also involved in this project. And I said, "Can you excuse me, please? I have to leave now because I think I may have won the Nobel Prize."
I'm sure they let you go.
They let me go, yes.
Okay, well, that's a brilliant story. Thank you for sharing it with us. So in the book, we just mentioned as we were chatting before we came live on air, you've got a wonderful example of a French scientist who I think is beheaded, and the judge really says, "We have no need for scientists here." This is a long time ago for those of you who haven't read the book. So a question from Caroline that kind of follows up on that or connects with that perhaps. What is your take on how the pandemic has been handled by the UK government, and how have the voices of scientists, unlike that French scientist, been listened to enough?
Oh, do you want a week of commentary? Handling the pandemic has been really difficult. I think the government has struggled. It made a real mess of testing, and it still isn't sorted. Vaccines done much better, so they have struggled. And I think one of the problems is that politicians are not used to having to convey difficult messages, and science is always difficult. And sometimes, in the case of a pandemic, it's only tentative knowledge. You're rather ignorant of the details of the virus and how it will be transmitted. And you remember we were told to wash our hands all the time. It doesn't transmit that way. We now know it's through the air and things of that sort. So it was complicated messages, yet politicians, and particularly maybe at this time with the populist bent, like one-liners, it's we are following the science is an example. What science are they following? Why are they following it? Is it likely to change? All of this loses the precision or the apparent certainty of the one-liners. One-liners win elections, like get Brexit done. I almost expected to hear get COVID done being said by one of them. Now, we didn't have that, but I think communication was not good. I think the testing was bad. I think the vaccination is pretty good. So mixed bag.
Okay. And I think one of the things that is clear from your book actually is that science is sort of messy and interconnected, and there isn't always a kind of direct route forward. And that's, I think, a lesson that we probably should all take on board. With that in mind, there's a couple of questions coming through about your own engagement still with research. So do you still find time for research?
Yes, to be quite honest, it's my major love. I run an institute. I'm sat in it here. It's a very big institute, the biggest biomedical institute under single roof in all of Europe. I've got 1,300 scientists here, more actually. So I do have that responsibility. But actually, my first love professionally is my own research group. So I have at this moment 5 PhD students and a couple of postdocs and 1 or 2 technicians, and I spend probably a third of my week with them. So I'm still doing it. And when I step down from running this behind me, I will probably do that full time. There's something fantastic about finding out things that nobody knew before. And there's much failure, but just occasionally you do discover something, and it can be completely new and important. And that is a great feeling. And what's even more extraordinary is that I've been paid to do it all my life, just to carry out my own curiosity. I mean, I couldn't imagine a better life, really. I mean, as long as you're tolerant of failure, it's great.
Fantastic. What a great answer. So another question really about research. Jonathan asks, do you think we will ever see a cure, and that's in inverted commas, for cancer?
Well, I've been on the fringes of cancer most of my life. I set up Cancer Research UK, that many of you may be familiar with, now nearly 20 years ago. And what we need to realize is that cancer isn't a single disease. It's probably up to 400 different diseases, certainly 200 to 300, because although the outcome has a commonality, that is, we're all made up of billions and billions of cells, and cancer's caused when that goes out of control. So cells reproduce all the time, which is my research topic. I mean, understanding that basic process. But the way that can come about, the way it can malfunction, is extremely diverse and different across different cancers, where they appear, what goes wrong. And each of those will need to be treated in certain different ways. So there'll never be a single cure, because it's not a single disease. It's several hundred diseases. And it is probable that it's safer to say we may never cure them all, but at least we should get ourselves where we can reduce a lot of the fear of cancer by being able to manage it, by keeping it under control, And gradually over the years, and I'm talking decades here, I do actually think that will happen. We now know enough on how to do it and how we could perhaps treat it for that to succeed. But there will never be a single magic bullet like penicillin. It's going to be hard work for decades, eating away at the many cancers and finding solutions to them.
Thank you. So time for one more question, and this is a really good one to finish up with, I think. So this is a question from Jenny, who says, do you have any words of wisdom for an aspiring scientist who is just beginning their career?
Oh dear, Jenny. I don't know if it's inspiring words. What I will tell you is you need to be driven by an insatiable curiosity of wanting to know the answers about questions concerning how the natural world and our cells work. It's overwhelming curiosity that drives discovery, and it's discovery that enhances our culture and civilization. If you like that, and you can cope with the fact that it doesn't work a lot of the time, then it is exactly the job for you.
Fantastic. And I think that lesson to all of us to be curious and take curiosity into the world with us is a really good message to finish on. So that's all we've got time for tonight. Thank you so much to our speaker this evening, Sir Paul Nurse, and thank you to you, our audience, for watching the 2021 Fickling Lecture on Developments in Children's Literature. The next Insights event will be on Thursday, the 11th of March. What's the Story? The Job of Directing Character-Led Action Films by Mark Jobst. We hope you'll join us next time. Thank you, Sir Paul.
Thank you, Jo.